Hard Science series. It's been fabulous. We've had amazing turnout and lots of great response. So I'm glad you guys are all here today to hear our two fabulous speakers. Our first speaker is Carmen Works from the chemistry department, and our second speaker, yay! And our second speaker is Shona Mookerjee from the biology department. Yay! Okay, oh my God, here's even more people. Come on in. And just a quick reminder, if you haven't had a chance to visit the art gallery yet, that exhibit is closing on the 31st, and that exhibit is called Women, Art, and Science. Okay, so without further ado, here's Carmen Works. Yay!
Okay, so my name's Carmen Works. I'm from the chemistry department. Most of you look familiar, so I believe most of you know me. But for those who don't, you can find me on the 3rd floor of Darwin Hall. So Karen asked me to give this talk and to talk about what I do as a scientist, and then also give you a little bit of background of how I got here or why I do what I do. And so I thought I'd start with this picture of chemistry. I see some people are chemists here, and others are not. And if you read any general chemistry textbooks, which are all pretty much the same, and you open them up, you'll have a definition of chemistry, which is the study of matter and how it changes. And I always like to tell people that the study of chemistry is the dance of the electrons, or the study of the electrons. That's kind of our focus of where we fit into our little scientific niche. And the chemistry itself, we break it down into 5 traditional subdisciplines. The first one of those, not listed in any order of importance, but for whatever reason, kind of the order in which we have students take these courses. And so if you get a degree in chemistry, or you talk to chemists, you'll talk about these different subdisciplines. And the first one is the analytical subdiscipline, or we like to call it anal chemistry. And it is just that, very precise and very rigid measurements. And I think sometimes when people think of chemists, that's how they think of them. It's very rigid and very precise. And I thought it was kind of cute, this little picture of a white guy in glasses and a lab coat, but a very precise measurement with a tie on. Because I think that's the picture we bring up a lot of times when we think about chemistry and chemistry. Then we also have organic chemistry. Now everybody loves organic chemistry. That's going to go, oh, I love organic chemistry. I love it. And you see in that, I don't know if I have a pointer, but I'll use my hands here. We also start introducing students to this thing that we call pushing the arrows. And that's where we push electrons around. So you can see them dance. It's the first time you see the dance with the electrons when you get to organic chemistry. And maybe that makes sense to students. And there's not a lot of math. So for whatever reason, even though our students are really good at math, they love organic chemistry where there's little math. They tell me that. So they'll go, oh, I love organic. There was no math. And then they go into physical chemistry, which is quite the opposite. There's a lot of math. You do a lot of calculus. But when I think of physical chemistry, I think of pictures like this that I'm showing you, where I think of calculations, or I think of looking at the energies of molecules in different states. Then biological chemistry is kind of an interesting one, and physical and biological chemistry are kind of on the edges of this spectrum, where you think of like, where's chemistry fit in terms of physics, right? And there's always this hierarchy of science that people talk about. They'll always talk about it. I don't think you can get away with it, so the physicists will say they're the hierarchy, they're on top, and then the chemists, and then the biologists. And then within chemistry, we'll have physics, and we'll have biochemistry, and they fit in this weird hierarchical thing, right? But the truth of the matter is all science is science, and all those lines are blurred. Not just between the disciplines of physical and life science, but also within these 5 subdisciplines. So biological chemistry, or biochemistry, gets its own. It's very interesting to me that most universities, we have a biochemistry degree. We have a chemistry degree and a biochemistry degree. We don't get an organic chemistry degree. We don't get a physical chemistry degree. Why that is, I don't necessarily know the answer to, but I always think it's interesting. So biochemists study biological molecules, or their biological relevance. And then there's the inorganic chemists. So the inorganic chemists, I have to mirror that with the organic chemists to put that into some type of perspective. If organic chemists study carbon molecules, specifically carbon-hydrogen molecules, there's a couple carbon molecules belong to the inorganic chemist. Then the inorganic chemists study everything else. There's no periodic table in this room, or maybe I should have brought one. But as you can imagine, there's a lot of other things besides carbon on that periodic table. So that's left to the inorganic chemist to take care of the rest of it. But there's a subdiscipline called organometallics, and then that pushes the organic part in there also. And I put a picture of these different colors because we often think of transition metal chemistry as being inorganic chemistry, and I like transition metal chemistry. One of the reasons is it gives all these beautiful different colored molecules. So I put that in there. So I'm like a mishmash between bio and inorganic, and I call myself a bio-inorganic chemist. And if you ask any of the chemists in our department, they will call themselves some kind of mishmash of this, right? So Jen would call herself a biophysical chemist. And Dr. Fukuda would probably call himself a physical organic chemist. And Dr. Perry would probably call himself an analytical physical chemist. Or maybe he would just call himself a physical chemist, I'm not sure. But he might call himself an environmental chemist. So that's not listed because it's not one of the 5 traditional subdisciplines. And then Dr. C would call himself a physical chemist. So biological chemistry or bioinorganic chemistry, what is that? So it is the study of inorganic molecules that are biologically relevant. My passion specifically are transition metals. But it doesn't have to be transition metals. But I'm going to give you a couple examples of those. So the first one here on the top is iron. And I think people can relate to iron because people know about anemia, if you're anemic or if you have an iron deficiency, so why you need iron. And so you need iron to transport the oxygen in your blood. And then you have iron in a molecule called hemoglobin, which is in your red blood cells. And copper is kind of an interesting one too. Arthropods use copper to transport the oxygen in their blood. And this is one of my favorite stories. So horseshoe crabs have blue blood because they have copper in their blood. And humans have red blood because we have iron. Or also, I think the Vulcans on Star Trek have copper in their blood. And they have a bluish green blood. So I always like that too, when the science goes over to the media and to science fiction. I like it when it's that one's good. That one's good. There's a couple other examples of some transition metals, and I point out this one of chromium because it's something that I spent a lot of time thinking about, and I'll talk a little bit about that today as well, since I'm talking about what I do. But chromium, so of all the first row transition metals, all of them are biologically important, except for titanium and scandium. But no one knows a lot about why chromium is important. So let's talk a little bit about that. So my research is in this area of bioinorganic chemistry, and I have 2 main topics. One is I look at chromium and the biological importance of chromium, and then also I look at some iron molecules that are important in catalysis, and I'll talk about each one of those projects. One thing I want to point out, though, while I'm on this slide, is this is a food label of nutritional values of stuff that's in your food. And you'll notice there's a lot of transition metals in there. And I circled the one that's chromium specifically because you have probably ingested a fair amount of chromium, but we don't know why you do that. We also know that it helps you lose weight, but we don't know why it does that. We know we prescribe it to diabetics, but we don't know why it does that. We also know that there's different forms of chromium. So we eat one form, but the other form we find toxic, like in that movie, Erin Brockovich, you know, where everybody had cancer because they were exposed to chromium. So just something to think about. So these are the things I think about all the time. So here's something that when I think about a problem, I think small. I think really small. So this circle, double circle, is my cartoon of a cell. And when I think about the chromium problem specifically, I think about 2 things. So the Erin Brockovich movie talked about hexavalent chromium. I don't know if you remember that, but that's chromium-6. And so chromium-6 is a pollutant in our water because it's highly water-soluble. So that makes it dangerous. It readily transverses these cell membranes. So not everything can get through your cell membrane, but chromium-6 can. Because it mimics other molecules, those ones that I have in the box, sulfate, phosphate. It mimics those. It looks like them. So it goes right in. And once it's in there, it goes through this really complicated redox process. where it changes. It changes from chromium-6 to chromium-3. And it can do that in 2 different ways. One of those ways, it gets stuck to your DNA, and that causes some problems, cancer being one of them. But it can also get reduced this way, and nobody knows a lot about what happens here. And so that's one of the problems that we're working on in my lab, and I see some of my research students, and I know it's a frustrating problem, it's a tough problem, but we think about that a lot when we work on it, and we try and isolate the molecules that are bound to that form of the chromium-3, the proteins and the peptides, and study them, and try and understand what's going on there. We also try and understand if you need them for this thing called glucose metabolism. So if you are type 2 diabetic, is it because you lack this chromium-3? And if you take chromium-3, we know it can't get into your cells, so this is really confusing to us. We don't know what happens to it, but for whatever reason you lose weight and your type 2 diabetic symptoms do reverse themselves. So interesting question, hard problem. But we chip away at it little bit by little bit every single day. The other problem that concerns chromium that I work on in my lab is I look at this toxic chromium-6 and I look at prokaryotic cells, specifically bacteria, that can maybe be useful in removing that from our environment in some way. And I have some students here that work on that project. Again, a challenging project that we chip away at every day. Okay, so the third project that's in my lab that I'm interested in is understanding how an enzyme called iron-only hydrogenase works on a molecular level. And so here's the active site of that enzyme. And so as an inorganic chemist, what I do is I peel away, these enzymes are these huge molecules, and I peel away until I just get the active site of it. And I try and figure out what's going on there. And in this project specifically, we make small models of the active site. And we use a tool called photochemistry, and we probe the reactivity of those small molecules to try and figure out what's happening in this larger molecule. And this is kind of neat because it catalyzes the reverse oxidation of molecular hydrogen, which is like this really fancy thing to say, but really what you're doing is taking H2 and breaking that bond, and then you can use those electrons for energy. So if we want to utilize hydrogen, we could make catalysts, well there are catalysts, that model this system that are cheap and could be easy to use. Right now we use platinum. Platinum's more expensive than iron. I probably didn't have to tell you that. But in case you didn't know, platinum's more expensive. So I got interested in this whole area of bioinorganic chemistry when I was in graduate school. So I just told you what I do. And those are the problems that I think about a lot. I also teach here. And how did I get there? So I'm going to go backwards for a little bit, and I'm going to start with nitric oxide. Because nitric oxide was the molecule of the year for a while when I was in grad school. It was all the thing. And so I wanted to work on nitric oxide. And I was interested in biological problems. And so nitric oxide is a small molecule that's biologically relevant. And one thing it does is it activates this enzyme called guanylyl cyclase. And guanylyl cyclase is an iron heme. So there's iron, transition metal, biological system, NO, I'm in, right? So here's the active site of this molecule. Again, it's a huge molecule, and I've pulled away all the pieces, and I'm just showing one very small piece. NO binds to that, and it causes a conformational change in the molecule, breaking that axial histidine, blah, 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 but there's a change. And when the molecule changes, it can then catalyze this reaction by binding that GTP. And that leads to smooth muscle relaxation. Viagra. Do I need to say more? So NO is important, and it relaxes your smooth muscles. NO does other things. It's also an important neurotransmitter. And it's also the first line of defense of your immune system. So your immune system comes in and it'll spray antigens with nitric oxide and kill them. And so when I was in graduate school, we were interested in all these different questions except for really the neurotransmission question. I don't think anybody's really studying that right now because maybe it's too, I don't know why. We could ask Dr. Fukuda about that. But we were interested in all these questions, and I wanted to get in there. And one of the things I wanted to do was make molecules that would release nitric oxide using photochemistry, and then maybe you could take those molecules, put them in tumorous tissues, and mimic that immune response and kill cancer, because everybody wants to cure cancer. Every scientist wants to cure cancer. I'll tell you that right now. I'm in graduate school, right? What do I want to do? I want to cure cancer. So I'm going to go along, and I'm going to make those molecules. So we were interested in two things. We used ruthenium, because ruthenium is a lot more stable than iron, and it'll also bind NO. So we were interested in understanding the photophysics of it, and this is a simplified picture of it, but here's a very simple ruthenium nitrosyl compound. You shine light on it, and it releases NO, and then we could try and use that as a tool to do different things with the nitric oxide. Interestingly enough, if you take ruthenium molecules and you react them with NO, they'll do the reverse process, and that has some interesting properties as well. So in graduate school, I studied both the photo release and what you would call the thermal reaction of NO with transition metal complexes, and I mostly worked on ruthenium. The molecule I specifically worked on was these different Salin-type molecules, and I put that in there because they have other uses. They're also used in catalysis. So that's kind of interesting. And so I've kind of come full circle where I've come back to doing photochemistry with that iron hydrogenase project and using that to probe the reactivity of these molecules. And I love doing that. I love working with small molecules. I thought when I graduated from graduate school, when I was done, I would wanted to work with proteins. It seemed like the hot topic, discussing. Everybody was working with proteins. The proteins are really difficult to work with. They're large molecules, they're complicated, they're messy. Small molecules, even though making them can be a challenge, they're small. They're small. They're small, and then you can apply them to other bigger problems. And so that's what I like to do. I like to look at the chemistry of these different molecules that are biologically relevant and try and figure out what's happening biologically with those models. So I do a little bit of both. I do some protein chemistry, I do some small model chemistry, and I do some photochemistry. But underlying all of it, I'm interested in what's happening on the molecular level. And so I'm really interested in picking out the details, the mechanisms. I want to study the mechanisms. Okay, so how did I get to graduate school? So I'm not in this picture, but this is my group in graduate school, and I love this picture because this is shows a lot of collaboration too that we have. So this is, I'll have to find a laser pointer here. I know there's one up here. This is my research advisor in graduate school, Dr. Peter Ford. He's a very famous chemist. And he collaborated a lot with this guy, Jean Facuto, who you may or may not know. So Ford and I had a lot of heart-to-heart talks. Usually they were heated discussions. In graduate school in chemistry, it's a weird place. You know, you're never told you're doing a good job. You're always just told what's not good. And it thickens your skin a lot. And it's an experience you have to go through. I've been through a lot of different things in my life, but the one thing that's hardened me more than anything is going through graduate school. You get yelled at a lot. You get degraded a lot. And I bring that up because when I was in graduate school, I remember thinking, God, it would be nice to have some positive reinforcement, like just a little tiny bit. And Ford would say, yeah, we don't do that here. Okay, that's fine. I also have to point out this guy, Ivan Lurkovich, because he was my postdoc. And so he worked really closely with me in the lab and taught me a lot of really great chemistry. So when you go to graduate school, you know, unlike here where you're coming to my office all the time asking me questions, it's like you wouldn't go ask Ford a question, like, because he would say, why are you bothering me with that question? Literally, quote, unquote, has said that several times. So you would go and find another graduate student, or hopefully you would find Ivan, who was like this wealth of information. So I have to point him out there too. So these are all these people that were interested in this nitric oxide game and studying nitric oxide and coming together and sharing all of their knowledge. And that was the one thing about graduate school when I got there. I'll tell you a little bit more about how I got there, which was a very crooked path. But when I got there, I questioned if I had done the right thing. Should I be here? What was I doing here? And the time that I knew I did the right thing was going to these meetings, these things we call group meetings, and discussing science. And it was something that I loved. And I was like, oh yeah, I'm here. I'm in the right place. I'm good. So there's a group of people, a group of scientists. And as an undergraduate, I went to San Francisco State University. And I worked for a woman called Jane Xiley. And when I met her, her name was Jane Crever. And so this is one of those stories right now, like, how did I get to graduate school and what were those life-turning events? And my undergraduate research mentor was one of those life-turning events. There were several, but she was one of them. I was a double major, so I worked in the Department of Psychology and the Department of Chemistry. It's chemistry and biochemistry there. And my undergraduate research project was to make the serenium acac carbonyl, which was really hard. So it was to do the synthesis of this, which was very air sensitive, and I had to use a lot of really nasty chemicals to do it, and then determine the pKa of that bridging hydrogen. So that was my research project, my undergraduate research project. And when I got to Jane, she was my undergraduate advisor. And I was getting advised on chemistry, and I was actually a biochemistry major. I was going to get a degree in biochemistry. And she was telling me this and that, and I was saying, well, I should do some undergraduate research. And she said, oh, you can join my lab. And I was like, no, you're an inorganic chemist. I want to do biochemistry, because that's the rage. Why would I join your lab? And then I walked away, and I thought about it, and I was like, well, that's dumb, because the biochemist, who is kind of the heavy hitter of the department, she ignored all my emails. So I went back, and I groveled, and I said, can I join your group? And it was the best thing I could have ever done, because when you do an undergraduate research project, it's not necessarily like, am I working on the most sexiest problem in the world, but all of the techniques and skills that you learn from doing that project and answering these unknown questions. And I got to learn air-sensitive synthesis, which if I hadn't joined her group, I wouldn't have known that I loved that. I loved it. And she would do these things that were badass. I'd never seen anybody do, right? She'd open up a thing of boiling ether and throw this thing called NaC in, which is an amalgam of sodium and potassium, highly explosive, and she'd be like, yeah, it's dry now. And she would just do it with this elegance. While I was working in her lab, I had some summer research experiences for minorities that I took advantage of. And well, so I worked, I think, 2 years for her and 2 summers. And while I was there one summer, Jane got breast cancer. And that was a really devastating thing for me. And Ushi Simonis took over her group. And so Ushi spent a lot of time with us in the summer, making sure, that we were getting results and doing what we needed to do. So that got me thinking just in terms about women in chemistry, and I think also watching Suzanne's talk kind of got me thinking about it. And then I was just curious, how many women do we have in chemistry? So here are 2 influential women in my life in chemistry. And then the first thing I popped up was just how many degrees, how many PhD degrees go to scientists versus everybody else. What I don't know is how many of these are social scientists and how many of these are physical and natural science. But what I do take out of this is that more PhDs go to scientists than non-scientists, which I thought was interesting. And then I wanted to know, what about the chemists, and where are the chemists in this picture? So if you were here for Suzanne's talk, you'll remember her saying that in most sciences what we've seen is that there's an increase of women in science, and that's occurred over so many years. So if you think about the 1970s, the bachelor's degree for women only represented 17% in chemistry, and we're half now of the bachelor degrees. So we're definitely 50% women coming out of undergraduate throughout the United States of America. And we're not doing so bad in the PhD area either. So we're up here about 37%. I couldn't find the newest data, but this gives you an idea of the trend at least. Things have changed since the 70s. Things have changed since Jane got her PhD. They're different, right? So you also have to adjust a little bit for citizens and non-citizens. But the other thing I started thinking about is how many people does this represent overall? And I put biology in there just to show you that chemistry is a little bit smaller. So chemistry is only 7% of the PhDs. And then of that 7%, about 37 or so are women. So just to give you an example. But anyways, the point here is things have changed. Women are in science. Different from, okay, so I only have a couple more minutes, so different from when Jane was here, or when Jane was in the Department of Chemistry and Biochemistry. So I get my PhD, I want to go into science, I want to be a professor, and I come here to Sonoma State University, and I join the department. And the department was in that 70s era. So the department started around 1965. I was the first hire in 18 years. I was the first woman to join the faculty here at Sonoma State. And these guys hired me. Now, Gene Schomburg, who's in the middle, he didn't look like that when I got here, so I can't find a more recent photo of him. But it was a very interesting department back then, which was 10 years ago. Doug Martin, who was the more recent hire before me, he was hired through education. So this department hadn't done RTP, I don't think ever, you know? So there were a lot of new things that I had to do. This guy here in the corner, Les Brooks, he didn't talk to me for like a year, maybe 2. I'd be like, hey, how's it going? Turns out that was probably a good thing. But here I am in my era, where we're like much larger percentage of the PhDs, the women, right? And I'm in the 70s department. So that was an interesting thing for me. And I'm out of time, so I won't tell you too many stories there. But it makes you think, like, how did I get here? So I'm going to show you one more picture because I'm a first-generation college student. I'm first in my family to go to college. I'm going to skip this slide, but it kind of shows you the correlation and the obstacles that I had to overcome. And this is a correlation of your mother's education versus how good you are in math. And specifically, I was interested in this rates and measurements. And this is 8th grade math. But I'll just tell you the short story of it, and that is you're at a disadvantage if you come from a poor, uneducated background, like I did. But here's my mom and my grandma. My grandma has an 8th grade education. My mom graduated from high school, so I'm not doing so bad there. My grandma was always interested in math, even though she never really applied it or was not formally educated. So I do think of my grandma a lot in terms of doing math problems with her. They were mostly adding up receipts from dinner and then figuring out the tip. But still, you know, or she taught me how to count change. And those little things that I came across. My mom, here I am as little with my cousin Stephanie, and I have to point her out because Stephanie's been there every step of the way. Stephanie never went to college, although she's also very smart and also a workaholic. So I find that interesting. And there's my mom and grandma again. So my mom, how do I say this? My mother is a free spirit. And there's both my grandparents that I wanted to point out. My grandma just actually passed away almost a year ago to this day. And so these role models, these people that are influential in my career or my life, never pushed me to do anything. And I'll go through this really quickly because that's kind of a point of my story is I never took high school chemistry. I never took an SAT. I barely graduated from high school. I was wild in high school. I was free to do whatever I wanted to do. I had no adult supervision. And I lived in a big city. Needless to say, if you watch the movie Kids, you have my teenage life right there. That's a really difficult movie for me to watch because I'm like, oh my God, can somebody do something about these kids? Could somebody have said to me, did you go to school today? Much less, did you have any homework, or should we do that at the kitchen table? And so my mom's friends especially, and I'll end it here, always ask me, like, what happened to you? What happened? Why are you still alive? First, they always say that to me. I can't believe you're still alive. I see them now. Oh, I can't believe you're still alive. Like, oh, great, thanks. What happened to you? Why did you change? What happened? People are always asking me that. You guys don't ask me that because you don't know where I came from. But people who knew me in high school always ask me that. And I'll tell you what changed because I started thinking about this for this talk. I was working at that dry cleaners. I didn't work 9 to 5. I worked 9:30 to 6 every single day. And I spent all my money on clothes and partying. And I did that for 2 years. And so my job was like white sweater, 1 blue shirt, spot-on collar. I did that every day for 2 years, and I think I just got bored. And so students will say to me, like, oh, I'm in college and I don't like it, and I wish I could do something else. I want to go to work. And I always think, yeah, okay, So I'll end it here because I think that's what changed me, but I had a lot of remediation to do. I hadn't gone to school really since 5th grade. And things are difficult for me to this day because of it. I didn't take calculus till I was 23 years old. And I graduated with a degree in chemistry and psychology at 27 and went to grad school. I had a lot of catching up to do. So I'm going to let the next speaker come up and end it there. And there's me and my mom. And I'll just end it with that. So thank you.
So we're going to do a quick switch over of the computers. And our next speaker is Shona Mookerjee from the Department of Commerce.
Yeah, I'm going to go the long way here.
And then we'll have questions at the end for both speakers.
So I'd just like to start by applauding Dr. Works' bravery in showing pictures of herself from high school. I'm not going to do that today. Okay, so, right, I'm Shona Mookerjee. I'm a lecturer here at Sonoma State, and in my other job, I'm a senior postdoctoral fellow at the Buck Institute for Age Research. Has anyone heard of the Buck Institute? Or yeah, so most of you know about it. So it's an institute devoted towards the study of basic aging mechanisms and aging-related disease. So those two areas dovetail quite nicely, which is why they decided to build a whole institution specifically to study these issues. And so I've been there for the past 5 years, and I'll talk a little bit about what I work on, but I wanted to start by talking about what I liked when I was a kid. Because like looking back, it's easy to find common themes, but at the time I was all over the place, right? Like I like ballet, I like soccer, I like animals, I like eating, there are things that every person likes. And so to go back and decide, this is when I started loving science is a really hard thing. But when I was a kid, I really loved this PBS television show called All Creatures Great and Small. And I was determined that I would be a veterinarian and work with horses and cows. We never had any pets when I was little, so this was sort of an unrealistic goal. But I wanted it anyway. And then we had this book that is difficult to see here, but it's called The Reader's Digest Guide to the Human Body, or the ABCs of the Human Body. And I read this book so many times. It's unbelievable. And they had pictures of cells and tissues and membranes and different body parts, and it was fascinating. And I think these two things are some of the real first indicators that I would really enjoy the career path that I've ended up on. So I work on things in cells. This is a slide from my graduate thesis talk when I was young and spry. But so cells have a lot of stuff in them. But what I did in grad school and what I do again now is to study a specific part of the cell called the mitochondria. And there are these little kidney bean things here. And one of the reasons they're so fascinating is because they allow people to use energy, right? Mitochondria are how you process your food into energy that your body uses. And that mechanism underlies so much of what we know about health and disease and various states of health and disease, that understanding how they work is crucial to understanding how we work. And so despite the fact that people have known about mitochondria for about 200 years, we still know relatively little about how they work on a biochemical and metabolic level. And so I started out working on their DNA and DNA repair, and now I work on a more metabolic aspect of their function. But this is just a picture of what they look like in a cell. So this is the prettiest picture I could find online. So this is an animal cell. You'll see that the mitochondria are stained in green, so they're all these little tubules, and the other stains don't matter as much, but they just show you the cell's nucleus and then other parts of the cytoplasm. So the mitochondria in a cell can adopt many different shapes and conformations. So in this particular cell, they look like tubules. Sometimes they look like little spots. They're described sometimes as stars in the night sky. They're very poetic. But again, they adopt. So they the other thing they do is move and change. So this network of mitochondria can fuse and come apart. It undergoes these cycles of fusion and fission, and it's now becoming more apparent that fusion and fission are crucial to proper mitochondrial function. So one of the things that we sort of did a side project on in grad school was looking at whether the DNA that was in mitochondria could influence how they came apart and stuck together again. I worked on the model system baker's yeast. So brewing and baking both use this type of yeast. And you'll see that these are stained for mitochondria as well. And when you grow, the nice thing about yeast is that they can ferment, which is why they're so great for brewing beer and wine. But they can also respire. So fermentation is the process by which you anaerobically metabolize food products, and then you spit out either alcohol if you're a yeast or lactate if you're a person. But yeast can also respire, so they can use their mitochondria. They pull in oxygen, they metabolize, they spit out water, but they also generate a chemical called ATP, and most of you have probably heard of ATP. It's the cell's currency. It's how the cell stores energy to use for other processes. So I think one of my motivators for wanting to become a scientist and wanting to learn about how these things worked is because we're faced with so much information, and if you don't know how to deal with the supplement aisle of your grocery store, it's really hard to figure out whether these things are real. Should you buy these things? Should you buy something called lifespan extension complex? Is that a thing? Does it actually work? And it sounds improbable, and it actually is rather improbable. But just the idea that this whole set of stuff is getting pushed on you every day. And most people don't understand mitochondria. I barely understand mitochondria. So the idea that information helps me evaluate what to do, I think, has been a strong motivator. So this is the mitochondrial genome. So you have your nuclear DNA, which is the half set that you inherit from each parent that gives rise to your 46 chromosomes. But you also inherit cytoplasmic DNA that is housed within your mitochondria. And this comes exclusively through your maternal line. So the mitochondrial DNA that you have is the mitochondrial DNA that your mom had, that your grandmother had, and so on and so forth. And there are a number of mutations to this genome that give rise to congenital human birth defects and disease syndromes. So on the outside here is the human mitochondrial genome, and on the outside is that of yeast. And the main thing to draw from this is that even though the sizes are quite different, the genes that they encode are almost identical. So in grad school, I worked on how this genome was repaired and replicated. And so there are a lot of interesting, outstanding questions about how this works and how they apply to disease. But using yeast, you can see really nicely how mutating different parts of the genome that disrupt functional repair complexes can interrupt how they grow. So this top panel shows different yeast groups diluted from left to right. And you can see that when they're on glucose, which they can ferment, it's a sugar, most of these mutants grow all right, or just fine. But when you switch them to a medium that they have to use respiration to survive on, a lot of these mutants fail to grow. And so we explored this question of what is it about these mutations that we made that prevent these cells from being able to use their mitochondria to make energy. And yeah, I'm not going to get too far into the specifics of that, but we can talk about it later if you'd like. Right, so mitochondria are these things. So mitochondria are really the batteries of the cell. They've been described as the battery of the cell, the powerhouse of the cell, the water wheel of the cell. All of these analogies suggest sort of a relatively representative whole where the mitochondria are where energy comes from. So food is the input and energy is the output. And mitochondria is the black box in which all that stuff takes place. And so what makes this magic happen is that mitochondria have two membranes. They have an outer membrane on the outside and an inner membrane, which is tightly folded. And so this increases the surface area over which the machinery that makes that charge of the battery, if you will, happens. So you have all these little pumping mechanisms that pump protons across the inner membrane. So you set up a gradient. You have a positive charge on one side and a negative charge on the other side. So a battery isn't the greatest analogy, but it helps you have a sense of what it is that's driving the mitochondria to make energy. They're charged by the food that you take in to be a battery that does that. So what I work on now are aspects of this process, which is also called oxidative phosphorylation. So this is a picture of the mitochondrial inner membrane only, and you can see, so these are all the pumps, complexes 1, 3, and 4, that push protons out of the matrix, which is this inner space here, into the intermembrane space. And then you can think of the outer membrane out here somewhere. So the crucial part of this is that you have a pumping mechanism that then allows the protons to accumulate outside. And of course, they want to flow back in. So thermodynamically, this is an unstable situation, right? The protons want to distribute equally across this membrane. So there's a driving force to allow them to come back into the mitochondrial matrix. And they typically do that through this complex called the ATP synthase, or complex 5. And the action of those protons flowing back into the matrix provides the energy that drives ATP synthesis. So to recap all of this, you have food intake that provides the power to pump protons outside of the matrix. Those protons flow back in, and that flow of energy allows the cell to synthesize ATP. And so one of the things I work on now is this idea of a decoupling shunt, if you will. So there are ways that protons can leak back into the matrix without making ATP. So you can have this non-productive cycle of food metabolism and equilibration of the protons. And so, I mean, you might think that that's a silly process. Why would your body want to do that? Well, in this day and age of excessive food and calorie consumption, a lot of people are very interested in ways to decouple food intake from energy synthesis and storage, right? So the idea that you can use uncoupling as a treatment for obesity and diabetes is currently huge. We're not particularly interested in all aspects of it, but it's something we think about. So the ways that protons leak back in are they can kind of wiggle across the membrane. There are a couple hypotheses for how that works, but there are also other proteins that can be activated to act as channels or pores. And so when activated, protons can leak back through those pores and therefore equilibrate the proton gradient. So these are called uncoupling proteins because they uncouple the process of oxidative phosphorylation from ATP synthesis. So most of you have heard of free radicals, right? You take antioxidants, you neutralize free radicals, your body is healthy and strong and stays young forever. And that's the idea. And part of the reason we care about uncoupling proteins is because they can help to minimize the production of free radicals by mitochondria. So what I forgot to mention back here is that this process is crucial for life, right? Your mitochondria have to work for you to live. But part of this process is that when the electrons flow down this potential chain, they can sometimes fall off and they get sucked up by oxygen, and that makes a superoxide free radical, which can then go on to react with other molecules and make other kinds of radicals and cause problems in your cells and in your body. So this is a chart showing you that relationship. So we have this force, the driving force of the battery. It's called proton motive force. And so as proton motive force goes up, the rate of free radical production, in this case superoxide production, increases exponentially, right? So there's a sweet spot in here where you have a high enough force, driving force, to make all the ATP that you need. And if that proton motive force is too high, you make a lot of free radicals. And so one idea about what the uncoupling proteins do is that they help keep the proton motive force low enough to maintain the ATP that your body needs without pushing you into a high force that makes a lot of free radicals. And so the idea, because we're in a lab that works at an aging institute, we always have to pull in ways to justify this research towards aging. So one of the models that we have is that you spend a lifetime fluctuating in your energy intake and energy demand. And so delta P represents the force that your mitochondria generate over time. And so typically, to maintain the best health over the longest period of time, you want to maintain ATP production and balance it against the PMF in a way that leaves you enough of both, right? You have enough ATP, you're not making too many free radicals. You can dip below this line and have insufficient ATP production, which can cause a number of diseases, but then you can also have too high a PMF that leads to high free radical production. So it doesn't have to be that this happens all the time, but if you have a lifetime history of enough rises into this area that make free radicals in these spurts that can then go on to have lasting damage in your tissues, this is thought to be one of the ways that free radicals might contribute to the aging process. And there are a lot of aging-related diseases that seem to speed up the aging process. And a lot of those diseases also are correlated with an increase in free radical production, or people can measure more free radicals in tissues of people with these diseases. So the idea, again, is that mitochondrial uncoupling will discharge this extra force, right? The force that you don't need to make ATP because you're already making enough. So this is just a flow chart showing that. And so the idea also that uncoupling proteins do is that it's a negative feedback loop, where if you make mitochondrial free radicals, or reactive oxygen species, they can cause oxidative damage. These radicals will attach to stuff and disrupt the chemicals and molecules in your cell. And so some of those products, those oxidized products, might activate the uncoupling proteins, which will then decouple respiration from ATP synthesis. And then you'll lower PMF and lower the rate of free radical production. So it's sort of like the thermostat in your house, the classic example of negative feedback, where if something is out of whack, you have a system to restore it to an equilibrium balance. And that's exactly what our model is for how these uncoupling proteins work in cells. And there's a lot of evidence that uncoupling proteins in particular can be important for maintaining health. There are a number of papers discussing the role of UCP2, uncoupling protein 2, in artery stiffening, in skeletal muscle function. Insulin resistance is a big thing now because it's the precursor to diabetes. So the idea that your glucose homeostasis physiologically can be dysregulated is thought to lead to a syndrome of symptoms, diabetes among them. And this requires a tight balance of glucose and fat metabolism, which UCP2 is thought to regulate. It's implicated in the development of cancer. So there's this idea that as cancer develops, the cells stop being respiration dependent and can rely mostly on glucose metabolism. So then people look for specific markers of metabolism that might indicate a cancerous cell. And the more pathogenic or progressive these cancers are, the more UCP2 they express. And then finally, uncoupling proteins have been implicated in lifespan itself. And so we recently reviewed a lot of the literature about whether it's true or whether it's believable that uncoupling can affect lifespan. And we were inconclusive in our answer, so don't ask me what we found. So the uncoupling proteins all look like this. This is the structure of uncoupling protein 2. So this is the mitochondrial inner membrane. So this is a protein that has 6 transmembrane spanning domains. They kind of fold in on themselves into a pore or channel. So this is the structure all spread out. And you can see it from the side here and then from the top looking down. So it forms a channel. And so one of the models for its action is that protons can flow through this channel when the protein is activated. And that's how it acts as an uncoupler. So you have a couple different uncoupling proteins in your body, and they're expressed in different parts of you, in this case, a mouse. So UCP1 is this classic brown fat thermogenic uncoupler. So babies and rodents are 2 organisms that have a hard time maintaining their heat balance. And so shivering sometimes doesn't always get the job done. So what they have this store of what's called brown fat as opposed to white fat, which is the fat that you normally think about. And they're brown because they have so many mitochondria. They're jam-packed with mitochondria. And they have so much UCP1 that this brown fat basically acts as a heat generator. So when you uncouple, the energy that doesn't go to making ATP has to go somewhere, so it gets released as heat. So your brown fat is a heat-generating tissue because of the high density of mitochondria and the high proportion of UCP1 that those mitochondria have. And so UCP2 and 3 are the other homologs that we study, and they're much lower in their expression, and they're much less extreme in the things that in the products of the uncoupling because of the low expression. And so UCP2 has been reported in the brain, the spleen, the lungs, the kidney, most importantly for us in the pancreas, where it's been shown to control insulin secretion in response to glucose. And the other really exciting location for one of these homologs is the skeletal muscle, because muscle is such a huge contribution to your whole body that if you can harness UCP3 for uncoupling, you can make large inroads into treating obesity, for example. So one of the things we look at is that is whether, because you think if you have a system like this, you'd have to control it really carefully. And so we look for ways that we can find that the body controls the activation and turning off of these proteins. So we know for UCP1 that there's a very clear biochemical acute pathway that happens within minutes, right? But for UCP2 and 3, that's not very clear. And so we've studied how these uncoupling proteins get turned off. And one of the ways we think that happens is by the proteins getting degraded by other stuff in the cell. So this is a Western blot. Many of my students have seen this many times. But these are actual proteins that have been targeted with an antibody and then visualized on a piece of film. So the intensity of the band represents the amount of stuff, the amount of that protein that's there. And you can see that over time we can make UCP3 go away, which means that its turnover is really rapid. So this protein is getting made and then degraded really quickly in this futile cycle. But we think that the reason for this is because it's getting turned on, and then the only way to turn it off is to get rid of it. And so that's one way that we've described how this works. And there are a couple of things here about how it works, but I'm not going to get into it. And the other thing that we discovered or found was that the driving force, the proton motive force, also controls the rate of turnover of these proteins. So this is the most important part of this figure, where you can see that at a high driving force, you have a high rate of turnover, which is why it's relatively low on this scale. And as you have less driving force, as you go backwards down this chart, you see that the rate of turnover is slower and slower. So we think that this is a way that mitochondria can prime themselves if there's ever a toxic event that releases a lot of free radicals. The proteins that are there turn on, but it also lowers the proton motive force enough that more of these proteins can build up in the membrane. So that if you have a situation where you have a highly fluctuating environment of energy demand and release, that you can change the structure of the mitochondrial membrane to be more adapted to dealing with that high fluctuation. And then over the course of the next couple days or weeks, it will return to its normal state. So we're exploring a system where you can have membrane level changes in the protein composition that affect how these cells can metabolize and how they can control their free radical production. So some of the questions that we're working on in the future are, of course, dealing with all the sexiest questions you can think of. Does UCP2 function in diabetes? Can you use it to maintain stem cells, as a side project that we've started working on recently? And can you use UCP2 as a biomarker of cancer? So these are all things that I have worked on or am working on currently, and it's really exciting. So we're hoping that it produces interesting results in the next year or two. And with that, I'll finish up and take questions. So I guess we're both taking questions now, right?
So anybody? For either of our speakers?
Yeah, I want to know what those favorite quotes were. Okay, well, I can't remember all of them, but I will say there's one quote that particularly stands out. One day, an adjunct professor walked up to me and said, "You know, you're really man-like." You think you could work on that? That was one of my favorites. And I didn't really know how to respond to that. Because I didn't think I was man-like. And I was like, do you mean the way I dress or my mannerisms? She's like, yeah, all of them.
question.
Was it a positive or negative?
I didn't know. That was the question, right? Was it positive or negative? So I'm like, well, maybe it's positive. Maybe I want to be more man-like. But I don't think it was. I don't know.
Because it could mean that you're assertive and confident.
What was that?
But
Positive
sometimes
or
it's
negative?
hard to tell.
It was hard to tell. The other one, administrator, oh, I know the speed tape, was, oh, that Carmen, you gotta watch her, she's got personality. That's one of my favorite ones.
Did your undergraduate advisor survive breast cancer?
No, she's not. She survived it the first time, but not the second time, not the recurrence.
Other question. On your last slide, you talked about new research that you're doing with stem cells. So, all that changed, right, with the Obama administration?
Well, so we're careful to work on NIH, so National Institute of Health has approved about 30 lines for research that they'll fund. And so we're very careful to use those cell lines and those cell lines only. And so California has this organization called the California Institute for Regenerative Medicine, or CIRM. And so we also have CIRM funding, but because we work in a lab that receives funding from both, we have to be careful, because if you work on a non-NIH approved line, none of the equipment, none of the personnel, none of the pipettes that you use can be purchased with NIH money. So we would have to essentially build 2 labs if we wanted to make our own stem cell line, for example. Yeah, so we're very careful to comply with federal regulations on our research.
We use EPC2 as kind of like an alternative treatment to insulin and diabetics, a way to regulate.
Exactly. So that's one of the great hopes about UCP2 research. So the uncoupling cycle will sort of dampen the insulin response, and so if you have beta cell exhaustion, for example, if your cells are so tired they can't make any more insulin, UCP2 is thought to be a way that it might restore some of that function. But it's tricky because, I mean, there's a positive and a negative to disrupting UCP2. So it's hard to tell right now whether the positive outweighs the negative effects on the cell also. But people are really interested in that idea.
Do you have to worry about as much your funding coming from different sources and?
Well, I always have to worry about funding.
Well, right, I
always
have to
worry
worry
about
about
funding. But no, not as much as with those types of regulations.
Yeah. And that's really stem cell specific.
You know, funding's already difficult to get, so
Yes.
adding that other layer of complexity.
Is there a question in the back?
I'm just curious whether you have some good recommendations for information for the layman who might not have the background you do. What would you suggest?
So depending on what you're looking for, a lot of universities will often have really good tutorials on like Huntington's disease or Parkinson's disease or Alzheimer's. There are a number, so I would go to a university website first before I went to like a pharmaceutical website. But the NIH also curates on their National Library of Medicine website a whole host of tutorials on various health issues, everything from arthritis to exercise to various diseases. And so that's also a really good resource for biological and health information.
And how about for the supplements? So I noticed you had one on there that was like mitochondria.
Yeah, that was one I thought was particularly amusing because the wrinkled mitochondria makes it old, and the smooth one is healthy and lively. So for supplements, it's really hard because most of the literature for them comes from the company that makes them. But there are papers available online that look at the efficacy of those treatments, but those are usually in the scientific literature and they're hard to grasp. There are a lot of blogs now where researchers go through and analyze papers or analyze issues and try to explore in sort of layman's terms whether or not these claims are true or whether they work. I can't recommend any specifics, but there's a lot of information now by people who are trying to interpret science to a broader audience, and I think that's a great use of the internet, in my opinion.
What advice would you give at this stages in your career
in graduate school, I met a lot of people who were there who didn't have a passion for it. They were there because they thought they were going to make money somehow.
Ha ha ha.
Yeah, exactly. And they struggled, you know, because lots of things to do.
I would add that having a passion is essential, but having, like, I was really lucky in having really good mentorship. I found an advisor that I got along really well with and was really excited, and I became excited then too about this topic. And so seeking out really good advice and mentorship, I think, is also key.
And I would add to that, mentorship, because I have a really, really great PhD advisor, but I didn't always get along with him.
Mm-hmm.
So
Yeah,
we
it's
weren't
not
always-
always buddy-buddy.
It's not always buddy-buddy. So if you, I think students get that idea, like, oh, we're going to have coffee, or we're going to hang out, we're going to, like, you know, go sailing and stuff. And
Right.
it's not like that. But that man will always have my back. Always. And I know that. And that's solid.
Mm-hmm.
So that's something I think that we get confused here. It's no mistake. When we are too buddy-buddy.
Wow.
Because humanity's down now.
So my intern is here, Fujin. She works for me at the Buck.
Shona, I have a question for you. I know you're not going to work for Buck forever.
That's true.
What is your future career objective?
That's an
Will you
excellent
consider working for a pharmaceutical company?
That's always an option. So I really enjoy teaching. I think teaching is really important, and I think people who really like teaching should try to pursue it if they can. And so that's one of the drivers for me to pursue a teaching career. But research is great. I love science too, and so I could see doing any number of things. But I think my first priority is to try to get a faculty position. But barring that, yeah, I think any research career, any editing career, anything related to science and the distribution of science is something I'm interested in doing.
So what about Research One? Like, you're working at UCSF, for example.
Yeah, I mean, so I think like my priority right now is sort of a teaching-heavy institution. I went to an undergraduate teaching college, didn't have a graduate program, and I thought that the focus on teaching was so great for me that I wanted to then propagate that. But that was before I started my research career, and so, yeah, so I really like both things now. But yeah, again, I think teaching is sort of underserved as an idea in science, and so I really want to work on developing that.
This is for the speaker. When you applied to graduate school, were you really specific in what you knew you wanted to study, or was it general interest in that field?
For me, it was general interest in that field. I think there's a lot of interesting problems out there, so sometimes it's hard for me to just work on one.
I found it really hard when I was looking at prospective PI's websites because they were so excited about it, and I was really having a hard time figuring out why they were so excited. Like, they would write on the websites, "I love mitochondria," and I would think, how can you possibly love this thing? I don't even know what it is. So, and when I was an undergrad, I was an art major for several years, and then I switched to biology at the very end. So I hadn't had a lot of undergraduate research experience, or any, really, and so it was very cryptic. And I think I picked a university that I'd gotten into as an undergrad, but there were at least one or two faculty that I thought were working on interesting sounding things, and so they were enough that I felt like I had some backup. So it was a little chancy, to be honest. But I think once you're there and in that environment, you slowly figure out what it is you need to do to get somewhere that you want to be. Yeah, so the answer is no, I didn't look for a specific thing.
Any questions?
All right, sounds good, thank you.
Yay!